For over a century, there has been a golden rule in biology, a line drawn in the sand that separated all life on Earth into two distinct camps. On one side, you had the eukaryotes. These are the complex cells that make up you, me, mushrooms, and maple trees. These cells are architectural mansions, filled with specialized rooms called organelles. You have the nucleus holding the DNA, the mitochondria generating power, and lysosomes handling waste. It is a system of organized complexity. On the other side of that line were the prokaryotes, the bacteria and archaea. For decades, we dismissed these organisms as the studio apartments of the biological world. We taught students that bacteria were essentially small, unstructured bags of enzymes. We believed they had no internal walls, no private rooms, and no sophisticated architecture. The prevailing dogma was that inside a bacterium, everything just floated freely in a chaotic chemical soup. But recently, that line in the sand didn't just blur, it was completely erased. Deep inside the body of a notorious agricultural pest, scientists found something that simply should exist. They found a bacterium named Candidatus Profftella armatura, and inside that bacterium, they found a secret that challenges everything we thought we knew about the evolution of life. They found a genuine, complex organelle. This is the story of Profftella's secret, a discovery that hints at the mechanical origins of our own cells and offers a strange new hope for saving the world's orange juice supply. To understand the magnitude of this discovery, we have to start with the unlikely vessel in which it was found, the Asian citrus psyllid. This tiny, sap-sucking insect is public enemy number one for citrus farmers globally. It is the vector for huanglongbing, also known as citrus greening disease, which turns fruit bitter and kills trees. But the psyllid is a biological Russian nesting doll. Inside the insect, there are symbionts, bacteria that have lived permanently inside the insect's body for millions of years, passed down from mother to offspring. One of these partners is Profftella. Microbiologists have known about Profftella for a while. Unlike pathogens that make the host sick, Profftella is a defensive symbiont. It is a mercenary. It pays rent to the insect by producing a powerful chemical weapon called diaphorin. This toxin shields the insect from predators like ladybugs and parasitic wasps. Because Profftella never leaves the insect, its genome has shrunk over millions of years. It has discarded the genes for its own cell wall and DNA repair, relying on the insect for survival. In exchange, it devotes a staggering 15% of its genetic code solely to producing this toxin. It was already a fascinating example of biological cooperation, but when researchers from Japan and Korea used advanced 3D electron microscopy to look inside the bacterium, the story changed. Bacterial cytoplasm is supposed to be a boring, granular slurry, but when the imaging team reconstructed Profftella, they found it was packed with massive geometric structures. Running the length of the bacterium were gigantic, bundled tubes. These weren't random clumps of protein. They were architectural marvels. To put the scale in perspective, if the bacterium were the size of a human, these tubes would be like carrying a bundle of telephone poles inside your body. They measured hundreds of micrometers in length and were composed of fibers twisted into a perfect right-handed helix. Unlike most biological structures that collapse in a vacuum, these tubes were diamond-hard and resilient. But the true shock came when they looked inside the tubes. They were hollow, and packed within that hollow space were ribosomes, the cellular machines that build proteins. This is the definition of an organelle, a specific, stable, protein-based compartment housing a specific molecular machine. The researchers had found a ribosome factory, or perhaps a toxin assembly line, organized into a physical structure inside a bacterium. This shattered the bag of enzymes dogma. Profftella wasn't just a soup, it was a factory floor with specialized machinery bolted to the ground. Why is this such a massive shift in our understanding? We have to look at how we define complex life. We traditionally taught that bacteria rely on diffusion. If a bacterium needs enzyme A to meet molecule B, it waits for them to randomly bump into each other. Eukaryotes, on the other hand, use compartmentalization. We put enzyme A and molecule B inside a small room to ensure they meet instantly. This efficiency is what allowed complex life to grow large. Profftella shows us that bacteria are doing this too. We are seeing a quiet revolution in microbiology. We used to think only eukaryotes had a cytoskeleton, but we now know bacteria have cousins of these structural proteins. We have seen tiny cages called carboxysomes in cyanobacteria and magnetosomes in magnetic bacteria. But Profftella's tubes are different. They are not just small storage cages or temporary wires. They are a permanent, massive scaffolding that occupies a huge percentage of the cell's volume. They blur the line between skeleton and organelle. This suggests the bacterium has evolved a dedicated physical infrastructure to manage its most important job, the industrial-scale production of the diaphorin toxin. This discovery forces us to revisit the endosymbiotic theory, famously championed by Lynn Margulis. We know that our mitochondria and chloroplasts were once free-living bacteria that were swallowed by a larger host. Over billions of years, they lost their independence and became organs of the cell. When we look at Profftella, we are likely watching this process in real time. It lives inside the insect, it cannot survive outside, and it provides a specific service. In many ways, Profftella is already a defensive organelle of the insect. But the discovery of the tubes adds a meta layer. We have a host insect containing a symbiont bacterium, which itself contains an organelle of tubes. It is a biological turducken. This raises a difficult question. Did our mitochondria once have structures like this? Before they became the smooth power plants we see in textbooks, did the ancestors of mitochondria have complex internal tubes that they simplified away over eons? Or is Profftella inventing a completely new kind of complexity? The presence of ribosomes inside the tubes is particularly telling. It suggests a level of spatial organization for protein synthesis that was thought to be unique to the eukaryotic rough endoplasmic reticulum. Profftella seems to have invented a bacterial version of the rough ER using protein tubes instead of lipid membranes. It is a striking example of convergent evolution, where life finds two completely different ways to solve the problem of how to build proteins quickly. So why does Profftella need these massive tubes? Evolution is a ruthless accountant. It does not spend energy on giant structures without a reason. There are two leading theories. The first is the toxin assembly line theory. Profftella's primary job is churning out diaphorin. This molecule is a polyketide, a complex chemical requiring a massive enzymatic assembly line. The ribosomes packed inside the tubes may be there to produce the necessary enzymes at a breakneck pace. By confining the ribosomes and enzymes into a narrow tube, the bacterium creates a reaction chamber where the concentration of ingredients is incredibly high. This makes the chemical reactions millions of times faster than if the ingredients were floating in the cytoplasm. The second theory is structural. Profftella cells are weirdly shaped. They are incredibly long and thread-like, sometimes over 100 micrometers long, which is gigantic for a bacterium. A cell that long and thin is fragile. The helical tubes may act as a rebar skeleton, providing the tensile strength needed to maintain this extreme shape, allowing the bacterium to thread itself through the insect's tissues without snapping. Most likely, the answer is both. Structure often follows function in biology. A structure evolved to hold the cell open became a convenient place to stash the protein-making machinery. While the evolutionary implications are thrilling, this discovery also offers a practical weapon. Citrus greening is an existential threat to the orange and lemon industry. We have struggled to control the psyllid because it resists pesticides, and killing it harms the environment. But Profftella reveals a weak point. The psyllid needs Profftella to survive natural predators, and Profftella needs these tubes to survive and produce the toxin. If we can design a molecule that specifically disrupts the protein fibers of these tubes, a tube-busting drug, we could sterilize the psyllid of its defensive symbiont. The drug wouldn't need to kill the insect directly, nor would it hurt bees or humans, because we don't have these specific bacterial tubes. It would simply strip the psyllid of its armor, allowing nature, in the form of ladybugs and wasps, to finish the job. This represents a new frontier in pest control, called symbiosis disruption. Instead of attacking the pest, we attack the partner that makes the pest dangerous. Ultimately, the discovery of the organelles within Candidatus Profftella armatura is a humbling reminder of the limits of our knowledge. For centuries, we looked at bacteria and saw simple dots. We assumed they were primitive because they didn't look like us. But complexity is not a monopoly of the eukaryote. In the dark, high-pressure environment of an insect's gut, a bacterium evolved a secret. It built a cathedral of protein ropes, a factory of twisted helices, and a fortress of ribosomes. Profftella teaches us that life, no matter how small, is a relentless innovator. It blurs the boundaries we try to draw. It suggests that the secret to the origin of our own complex cells might not be a singular miraculous event, but a common tendency of life to compartmentalize, organize, and build. As we peer deeper into the microbial world, we are likely to find that Profftella is not an exception, but a pioneer. The secret is out. There is no such thing as a simple cell.